Good evening, everybody. Myself, Sharvari Kulkarni, and I'm Technical Associate at BDG Lifesciences. First of all, I welcome you all with a great pleasure on this webinar on bioethics and biosafety, walking a tightrope, bioethics and biosafety. I really appreciate that you have given valuable time for this session, and I assure that these 2 hours or this 1.5 hour will be really productive for you and informative for you. So I will start with the information which I have, and this topic is like bioethics and biosafety is like it's it would be more interesting if it would be two-way talks. So I will discuss whatever knowledge I have, and you also have a freedom to discuss with me whatever queries you have. So let us begin with uh, bioethics and biosafety. First of all, I will give you brief introduction of mine. I am Sharvari and I am Technical Associate at BDG Lifesciences. I have 12 years of experience in the field of biotechnology, and I have been teaching for Engineering College for Biotech Department for 12 years. And my areas of expertise include bioinformatics, intellectual property rights, bioethics and biosafety, and immunology, immunotechnology. So I have taught IPR Bioethics Biosafety for 12 years. I have done some courses of World Intellectual Property Organizations for this. And I used to be an invited speaker for entrepreneurship program which are conducted for the subject of intellectual property rights, bioethics and biosafety. Now I'm associated with BDG Life Sciences, and my area of expertise include bioinformatics as well. So we conduct trainings and workshops for all the subjects which are related to biotechnology, biopharmaceuticals, and bioinformatics. So today I'm really, it's indeed a great pleasure because this topic is very interesting, and whatever knowledge I have, I would like to share with you. So I would like to go ahead with a company's introduction as well in brief. I'll start sharing my screen. So let us begin with company's introduction. We are Biodiscovery Group of Life Sciences with 10 years of excellent service in biotechnology. Motive of our organization is to create a group of researchers, students, scientists, professionals, and whoever are related to the field of biotechnology, bioinformatics, and pharmaceuticals. So our group has free consultation, tutorials, books, project reports, assignments, trainings, job offers in India and abroad, scholarships, research news, latest science news, and much more. This is brief about the company. It's Biodiscovery Group, which started with in 2010, and we have wide array of services which include research projects, workshops, online on-site, training online on-site, webinars, internships, contract research, seminars, and conferences. These are some services which we offer on bioinformatics basics and advanced, on structural bioinformatics, on gene and genome editing using CRISPR, molecular dynamics and simulations, immunoinformatics, NGS, MD simulations, then R programming. These are some important services that we offer. I would like to tell you that uh, our team will put the links on the chat box. Please refer to that, and please visit our website to know the services which we offer and get the benefit of it. We would be also glad to offer you the services with best benefit. Our whole purpose is to make use of our knowledge for the fraternity of biotechnology. So I will start with the main topic now, bioethics and biosafety. First, I have divided this webinar into two important parts. First, we'll discuss bioethics. Then we'll discuss 2-3 case studies to make it more interesting. And then I will discuss biosafety cabinets. And at the end, I'll give you one quiz to make it more interactive. So first start with bioethics. Uh, what is bioethics? So I would like to divide this term into two parts. Bioethics, bio and ethics. First, bio and ethics means what? Science. Bio is science and ethics. Science says, could it be done? And ethics says, should it be done? So important that both are being considered. Science without ethics would be immoral, and ethics without science would be impossible. So that is how bioethics come into picture. Could it be done is the one question. Should it be done is the another question. So golden mean of both of this is following bioethics. So these are some contents that we are going to discuss. First, we'll start with introduction. Then we'll discuss what is bioethics. Then four principles which govern bioethics, which are very, very important. Then we'll discuss scope of bioethics in terms of latest technologies which are coming up. Then we'll discuss some issues or cases. And then we will come up to the analysis. So let us start with introduction of bioethics. It is basically the study of ethical issues which are emerging, study of ethical issues which are emerging due to advances in biology and medicines. So why are we considering bioethics? Is of course, if any technology is getting developed, there will be pros and cons of this technology. So there are ethical issues which we were not aware before development of technology. Now, because of the increasing or emerging advances in biology and medicine, we are becoming aware of what ethical issues are there with respect to this technology. So this bioethicists basically, they are concerned with ethical questions. What are these ethical questions related to? Life sciences, biotech, politics, laws, and philosophy. So it includes all the branches of medicines. Also, it establishes a common ground for different views, the democratic process being facilitated by principles of giving equal and fair consideration to all the sides or aspects of a given issue, with respect to environment, with respect to human, everything. So these are four principles I would like to talk about. First is autonomy, second is beneficence, third is non-maleficence, and fourth is justice. Autonomy. Autonomy is the rights of the individual to make decisions regarding their own health must be respected. Means whenever you are treating a person, it is the right of individual to make the decisions. Okay, so treatment will be with respect to doctors as well as individuals' willingness to go for the treatment. That is called as autonomy. Second is beneficence. Beneficence means healthcare providers, they must strive to the benefit of patients. So sometimes it becomes impossible to go with the willingness of the patients. There are some cases where doctors cannot listen to patients' willingness. At time, beneficence principle is followed. That means ultimate goal of the treatment is to benefit the patient. Third is non-maleficence. Non-maleficence is healthcare providers, they must not hurt their patients. sometimes this healthcare providers, they have to take decisions according to the situation. So, like for example, some unexpected complications which come during the surgeries. Okay, so that time, like shoot up of the BP, like that. So that time, so healthcare professionals, they have to take care of the patient's survival. So they have to give a priority, decide the thing, whether to follow the treatment or what is the compromise. Like if the side effect is more, then to stop the treatment, is it wise, or to continue the treatment if the side effect can be compromised? So that is called as non-beneficence. That means ultimate goal is not to hurt the patient. Justice. Justice means patient should get more or less equal treatment. That means every patient should get same line of treatment. That is known as justice. Okay. Now this we are going to discuss with the scope of bioethics. Scope of bioethics has expanded to include the ethical questions which are raised by Human Genome Project, stem cell research, artificial technologies like artificial transplantation technologies, organ transplantation technologies, possibility of successful reproductive cloning, pre-implantation. Sorry, first I should go up with the fourth point: pre-implantation of genetic diagnosis, synthesis of new life forms, possibility of successful reproductive cloning. Then first, there are five major issues that we are going to discuss with the bioethics point of view. First, abortion. Second, euthanasia. Third, eugenics. Fourth, human cloning. And last is stem cell research. So let us discuss one by one. And then at the end, I would like to know your views also on this. Yes. So basically, what do you understand by abortion? Abortion is basically it is a removal of fetus or embryo from the uterus of woman. It is termination of pregnancy. So the controversy, it is a very controversial topic actually. Controversy over abortion is actually a great debate over when life truly begins. So always, whenever you are talking about any ethical issue, there are two sides. So we are going to call in terms of bioethics these two these two parties as pro-choice bioethicists and anti-choice supporters. That means those who are supporting one view of bioethics and another who are opponents of this view. So pro-choice bioethicists, they place emphasis on how do they justify this abortion is they say it is entirely an ability of woman to have power over her own body. So no one else can decide whether she should go under abortion or not. However, anti-choice supporters, they say that life begins when it is conception. So you don't have right to end any sacred life. So there are two views. As the public and political debates over abortion are overwhelmed by religious belief. Religious beliefs means, of course, that they support you can't, life is given by God, and you can't end the life just because there are some complications and just because it is against the willingness of mother. So we understand there are two views. First, one. Woman will decide whether she has a power to undergo motherhood or not. It's entirely her decision. This is what is said by pro-choice bioethicists. And anti-choice, they say, no, it is a religious thing and you can't end the life which is given by God. So, bioethicists are kind of secular. They are they merely view fetus as a human life. Then next come euthanasia. What is euthanasia? Euthanasia refers to the act of purposefully ending life to eliminate untreatable sufferings. So this is a little new thing which has come up. Euthanasia means, in short, we also call it as mercy killing. So when there is a question of whether the life is worth preserving or not, and what lengths should be taken to preserve the said life. Supporters of euthanasia. So, as I told you, there are again two views, supporters and opponents. So, what is the claim or what is the justification of supporter? Supporter of euthanasia, they view the this course of action of euthanasia as a relief to unnecessary suffering if a patient is going to succumb to an incurable disease either way. That means, how do they justify this? Anyways, after giving a treatment, the period of suffering is also harmful to a patient, and anyways, it is going to succumb to an incurable disease. So, what is the point? It's better that if the person is not willing, let him undergo peaceful end. But opponents, again, they justify this with a religious belief that life is a sacred, it is a God gift, human life is a God gift, it is sacred, and it can fear a slippery slope into allowing treatable patients to die on their own will. So, if the patient he does not want to leave, there is no point in forcing a life. Anyways, it is going to end in the complication and death. Then why to suffer? Why it is uh, forcefully? Uh, why it is forcefully emphasized on the patient to live a painful life? So that is another thing. Then third, eugenics. Eugenics is basically a type of DNA manipulation that creates selective breeding to improve the human race. Human race meaning race is basically a species or class or a taxonomy. So, in order to improve the human race, DNA manipulations are done. I believe all of you are from life sciences background, biology background. So, you know what is DNA manipulation. Our genetic inheritance is done by DNA, and manipulating the DNA for better purposes is nothing but DNA manipulation. So, eugenics is transformation of this DNA for betterment. So there are again two views. Bioethicists who support gene selection, they obviously want to see the advancement of the species along with the extension of certain genetic diseases. So what is the purpose of this eugenics is to completely or the extinction of certain genetic diseases and betterment of the DNA. However, critics are worried about the slippery slope again that would occur. What is slippery slope? That means certain uh, unwanted or hazardous complications which come at the end of any experiment. So, since the eugenics have one more important point here is since eugenics has been associated with and discredited by Nazis' radical experiments to achieve an Aryan race. So there is a history behind this eugenics opposition, and that is Nazis' radical experiments. So what happened during this survey or study is some species of Nazis were extincted some features like tallness, fairness, then curly hair. These are some characteristics of Nazi species. And over a time, these characters exchange after DNA manipulation. So that is why opponents worry that when you are manipulating one race for a betterment, that race will be completely deemed inferior and eradicated. So this, of course, is an extreme argument for them. Why? Because they say that opponents debate that eugenics or DNA manipulations could lead to discrimination against certain races, disabled individuals, or even hair color, like changes in the characteristics. So that is why changes in breed or changes in the characteristics is opposed for DNA manipulation. Now, again, that is what bioethics is like, a questioning and answering. What is ethical? So, do you want to compromise over extinction of one breed for betterment, or do you want to do? You don't want to do it because it will extinct completely one breed. Next, human cloning. You all must have come across this term. I'm very sure. What is human cloning? Basically, it is the scientific production of an identical human cell, tissue, or entire body. So we are making clone or a Xerox kind of thing for the human cell, tissue, or entire body. Bioethicists that support human cloning see the benefits of regenerative medicine in terms of organ or tissue donation. Again, we are talking about two views. Bioethicists that support human cloning, they see the benefits of this regenerative medicine in terms of organ or tissue donation. So this would what would be the benefit of cloning? This would eliminate risk of body rejecting a transplant and the issue of scarcity of organs and tissues to be donated. So cloning has of course got a lot of importance because it has eliminated many problems that come when we have alternates of getting the organs or transplantation. But critics fear that the process will produce an onslaught of severely disabled children. This is also true, right? Cloning will ease many factors, will ease many complications, but there is another possibility also. Clones are produced, and these clones, because the cloning process is not like 100% guarantee, it is not standardized, it is not perfected. So there is a fear that onslaught of severely disabled children can arise. So always there is a risk. So it is nothing but this cloning for human body parts or vital organs is not advisable according to critics because it can produce human farming and killing of clones for their vital organs. Next is stem cell research. What is stem cell? Stem cells are the cells which are capable of multiplying themselves through mitotic cell division. I believe you all are aware of cell division, mitosis and meiosis, right? So these cells, they divide through mitotic cell division, and embryonic stem cells, we know that they also have the ability to differentiate into range of specialized cell types. So that is why stem cell research has incredible importance in the disease research. Controversy surrounds the use of these embryonic cells. Now, what is the idea or what is the justification of supporter and opponent? We will come to that again. Supporters see the potential that this research has in curing diseases such as Parkinson's disease. I don't know if you all are aware or not, but Parkinson's disease has got a lot of success with the treatment with stem cell research. You can go through the papers which are available to it. But again, when stem cells in the beginning, it was very supported. That technology was on book. But then critics started saying that embryos, basically for the stem cells, embryos are used for research which are left over from IVF treatments, and they are on the way to being destroyed anyway. So they are used. But opponents, they say that embryos are as the potential for new life again. Again, they have a claim or justification that embryos are also new life. So destruction of the embryos is again not justified. Okay. Now, I would like to discuss one very interesting case study. Yes. See here, this picture I would like to show you. I can see there are 7 participants. So, if anybody has something to talk about this case study or your analysis, I would be happy to know some input from your side or response from your side. I'll first tell you what is the case or what is the example that I would like to discuss. Then you can unmute yourself and give me the answer when I say. Okay. So, suppose here in the picture you can see there is a trolley which is running out of control down a track. In its path, you can say there are 5 people who have been tied to the track. Fortunately, you can flip a switch which lead to a trolley down a different track to safety. Can you see here? Yes. So unfortunately, there is a single person who is tied to that track. You're getting me? We have one track. Trolley is running out of control down a track. And in this path, we have 5 people who have been tied to the track. Fortunately, we can, since we don't want them to die, we can flip the switch. But on another track, there is another person sleeping. So should you flip the switch? Should you flip the switch? Same way, let me continue. Then you can maybe think for 5 minutes, 2-3 minutes, and let me know the answer. Okay. So a physician has this similar thing. I'm going to relate it to one case where physician has 5 patients, each in need, each in need of different organ, and will die without that organ. Unfortunately, there are no organs available to perform any of these 5 transplant operations. Now, a young man who just got hit by the trolley is brought into the ER emergency room. In the course of assessing injuries which are not life-threatening, if treated immediately, doctor discovers that his organs are compatible with 5 of his dying patients. Would it be justified for the physician to let the patient get or harvest his organs, or to save other 5 patients in order to save other 5 patients? Would it be justifiable for the physician to let the one patient die for the purpose of solving or purpose of getting lives to 5 patients? You understood my question? So anybody would like to give input? My team, I would like you to tell to unmute the person who wants to talk, please. Anybody spontaneously?
Ma'am, I could unmute
Yeah,
myself.
yeah, sure.
My answer is no for both the scenarios because
Both
there is
is,
no consent.
there is no consent, exactly. Okay, but who is this speaking, Nano? Your good name?
Ma'am, good evening. It's Neeraj Paliwal here.
Yeah, Neeraj. Okay, very nice that you thought in this way. But Neeraj, tell me, okay, this actually goes against the consent. Consent is not taken. Perfect answer. But then, if you are a doctor, what would you do
I will
in this
seek for
case?
the consent from the patient. And
Hmm?
my as my profession, if I am a doctor, my profession states that I must save the patient's life. So yes, on one table there is one patient, but on
Hmm?
another five tables there are five.
Hmm?
If the one patient does not allow me to take out his life and his organs to be donated, I do not have any right
Hmm?
to do anything.
Super. Very
I
good.
must perform my duties.
Perfect. Yeah. Fantastic. So here, excellent. I got response from your side. Thanks a lot, Neeraj. You
Thank you,
can
ma'am.
mute.
Thank you for the opportunity, ma'am. Thank you.
So I am glad that you responded. Okay, thank you, Neeraj. So you can mute yourself. Now, I would like to give you two highlights of the facts that Singapore has 7% GST and people get free medical treatment. But Indians, they do not get free treatments in spite of having 28% GST. Why? This again comes in ethics. So here, unfortunately, in India, records are seen such that bad records are seen such that oxygen supply cutoff is done if there is unpaid bill. On an average, 64 children they have seen to see the death because of unpaid bills. Sometimes doctor they just blindly switch off the oxygen supply. Is it ethical? Of course not. These are some things that we should give it a thought. Okay. Now I would like to discuss. This is one simple example that I discussed in the beginning to make it interesting. After discussion of biosafety, I am going to after biosafety discussion, I am going to keep 15 minutes session like this. If you respond, it would be very interesting. I have chosen two case studies of ethical issues and I would like to know your opinion. But since it would require some time, let me complete the program designed, and then at the end we will discuss it. Okay. So now I would like to discuss some issues of bioethics which are related to COVID-2. What ethical principle guides us in this public health crisis? So, as you all are aware, we had never seen, our generation had never seen such a critical or crisis like a corona pandemic. My grandparents and all, they say this was far in the past, during some pandemics of some plague and all. After that, this much bad scenario we have never seen. So, what ethical? So, people are like, suddenly we had to take up this challenge of fighting with Corona, lockdown, and everything we had never expected to happen. But now we have to follow other treatment as well as it is necessary, or it is a big challenge in front of healthcare professionals to make a perfect balance between the treatment as well as the ethical principles. So what are some ethical highlights that people are thinking that I would like to give you information about it? So ethical principles are fairness. As long as possible, you become or be fair to the affected communities, practitioners, and provider organizations. Duty to care. Standards are focused on the duty of healthcare professionals to care for patients' medical care. Transparency, duty to steward resources. Resources should be made available. Transparency in design, decision making, and information sharing. There should be transparency. Okay, we hear this news also in between, right? Some corona cases in some Gujarat and all some area, corona cases were hidden. Okay, so those kind of things are not advisable. You have to be transparent in decision making also, in data, what you can say, revealing also, and in information sharing also. Consistency in the resources and in the availability of the treatment. Proportionality, public and individual requirements must be proportionate to all the levels of the patients. Accountability of the decisions. These are, what do I mean by transparency? Nowadays, because people are also getting aware, then they asked government to be a transparent about COVID-19 updates. So these are all links that are provided. You have access to these links and you get data of all the COVID-19 updates. This is with the view of making people aware of exact status of corona. So not to get panicked, and at the same time you should know what is happening. So for that UW, that means University of Washington, they have or they are really focused on these views of getting the transparency in the COVID-19. Now there are some two important points that are need to be considered. HCP's duty to care. HCPs meaning healthcare professionals. Now, one question has been raised with respect to bioethics in this. Why do we expect healthcare professionals and first responders to risk their health and lives in this pandemic? They have families too. Yes, it is right. They have also families. This is also a raised question. Okay, but when you come across this question, uh, it's the fact that we have to accept, because healthcare professionals' job are not replaceable. It is not transferable. They cannot be easily replaced by others who lack appropriate training. Okay, so it is not like a computer training or something like that. You can get it trained soon and you can get the job. It's not like that. There is a big responsibility on the heads of healthcare professionals. So it is not replaceable, and it's actually healthcare professionals, they are voluntarily, they have accepted this amount of risk, knowing that they have to stand there when there is a case of emergency and pandemics. So they kind of take a promise or oath to serve in the pandemic situation. Everything is true, said and done, that they have families, but they have a duty to care. So here we can't raise this question with respect to ethics and non-ethics. It's basically a part of duty. Next question, duty to, next ethical issue is duty to protect HCPs. Yeah, this indeed is true. So HCPs are on their toes to help us. So it's our duty or it's government's duty to protect HCPs. So UW Medicine, that is University of Washington Medicine, they have a reciprocal duty to protect HCPs and keep them safe to the best of their ability. Okay, so you can hear nowadays that we also understand their uh, like what sacrifice they are doing for us, and we should be grateful, and also we should cooperate with them, and we should follow some SOPs which are given to us by government, given by WHO, in order to protect HCPs and also to lessen burden on this healthcare professionals. Now, sometimes with the ethical context itself, what is the say of HCPs? Healthcare professionals nowadays, they are saying that we are getting a pressure. We feel like we are abandoning patients when we just have to restrict visitors. That means visitors keep on cribbing that. We feel that you are not allowing us to meet our patient, so we get a pressure of abandoning patients. But you also think, we also understand that this is something which is not abandonment, but it is only the purpose is to prevent the spread of deadly infection. We are not freely choosing to restrict visitors. So patient and staff safety is required. It is the top priority rather than relationships, bondings, and love, right? So if we cooperate, that H cooperate, the group of HCPs, they should not get the guilty feeling of abandoning patients. So they also say that we also understand, but as the situation demands, we will safely relax the restriction, and they will. So we also should understand, cooperate, and prioritize the things. There is one more challenge which has been seen, that what happens when we run out of staff, ICU beds, or ventilators. So these kind of guidelines we get from UW, University of Washington Medicine, that resources should be shared within the region. So that is why you must have observed private hospitals also, they become COVID centers, because there are in a comparison with the population, with the great speed of spread of the disease, government will shortfall with the facilities which are given. So in these cases, resources will be shared by other hospitals, other doctors. They also should understand their duties, and they should cooperate with the government or WHO in order to facilitate for more ICU beds, staffs, and ventilators. Okay. So these were some ethical issues. First, I discussed bioethics in general. Then we discussed one simple case study, and then we discussed it with respect to Corona status right now. And as I told you, now I would like to go ahead with the biosafety information, and then we will come back to two case studies and quiz for bioethics. Okay. Yes. Now I would like to talk about biosafety. Biosafety is used to contain the spread of pathogens and toxins. It is implemented to safeguard the public from being infected. India follows World Health Organization standards with respect to biosafety. These are four levels of biosafety: BSL 1, BSL 2, BSL 3, and BSL 4. Yes. So this is a picture. I think most of you must have worked also in biosafety levels. So we are going to first see what are biosafety levels, and then we'll see what kind of biosafety cabinets do we have. So first is BSL 1. It is basic teaching laboratory. This is how it looks like. BSL 2. BSL 1 is basically your teaching organizations, teaching laboratories, institutions, colleges, where you have simple BSL 1. Then BSL 2. BSL 2 offers diagnostic services. Okay, diagnostic means some dealing with the pathogens which can cause infections. BSL 3. BSL 3 offers high containment spatial diagnostic services. So going up one level, BSL 3, they deal with the microorganisms which can cause little severe infections. BSL 4. BSL 4 going highest level up, that is, they handle dangerous pathogens. We are going to see all with examples. So these are the pictures. BSL 2, you can see here they are working in LF. BSL 3, BSL 4. So there are certain changes that we are going to discuss as in the upcoming slides. Yes, so this is a kind of pyramid that is constructed with respect to biosafety levels. First, you have BSL 1, which is non-pathogenic microorganism. It is minimum risk. It do not need any special containments. Simple tabletop arrangement will also do. One table, one burner, one uh, per, beaker is shown in this. No hazardous, simple working. So example of which is non-pathogenic strain of E. coli. So you must have all deal with this in your college labs also, in your college lab microbiome experiments also. So there is no harm. It is just for you to understand the basics of microbiology. So what are the basic safety practices, but of course they are not harmful, but they are strains of bacteria. So you have to follow certain basic safety practices like hand washing, wearing a lab coat, gloves, eye protection, limited access to people, no mouth pipetting, cleaning and decontamination of area. So these are some basic practices that you have to follow. So this is how it is. As I told you, these are some basic practices which we follow in your microbiology labs. Then comes biosafety level 2. Biosafety level 2 cause diseases in human beings. So pathogenic and infectious diseases. Going up one level, you are dealing with pathogens which can cause diseases or infections in humans. So biosafety level 2 is greater than biosafety level 1. How? If you have biosafety level 1 and you add on some things to biosafety level 1 standard, that becomes biosafety level 2. So first, using biosafety cabinet, it's not a simple tabletop arrangement, it's biosafety cabinet, trained personnel. So trained personnel is needed in order to work in BSL 2. Availability of IVOR station. IVOR station is always used in BSL 2, and vaccination for the workers. So it's always better to get vaccinated to a trained personnel who's going to work in BSL 2. Yeah, BSL 3 that cause serious or fatal diseases. So BSL 3 is one step above BSL 2. So here we are dealing with serious or fatal diseases like tuberculosis. Now here when we are talking about biosafety level 3, here comes interference of government agencies, medical surveillance till biosafety level 2. These all agencies or medical surveillance do not come into picture. But when you are dealing with yellow fever and tuberculosis bacteria like pathogens, that time government agencies as well as medical surveillance comes into picture. So you have to strictly follow the SOPs, and those are routinely regulated and they are governed or they are observed by medical surveillance team. This is BSL 3 facility where you essentially have HEPA filters to work. We are going to discuss what are HEPA filters in upcoming slide as well. Then comes biosafety level 4. Biosafety level 4 is rare in the world. BSL 4 consists of dealing with highly hazardous microorganisms like Ebola virus and nowadays coronavirus. So COVID virus, coronavirus is included in fourth biosafety level. So that is why it's difficult to have a setup to work or to research for coronavirus. So that is the big challenge. Okay. And what is the important factor or what is the important challenge in front of study for fourth level of biosafety is these are fatal infections and also they do not have vaccines. That is the first part. BSL 4 facilities, so you have to be at most care when you are talking about BSL 4 facility. You have supply specialized design of biosafety cabinets where you have supply air with HEPA filters, you have exhaust air, and everything is super organized and at super precaution, you can say. What are the laboratory practices of fourth level? Decontamination of materials before leaving, full body garment. You must have heard that all these practitioners they have to wear PP suits, right? So this is a full body garment, which is specialized suit, so that there is no what you can say microorganisms will not stay on the suit. Those are under pressure. These are specially designed. Then you have to take a shower after leaving the facility before entering the facility. All those SOPs are mandatory. Then we are going to discuss about biosafety cabinets. Okay, we are going to discuss little bit technical part of it, how biosafety cabinets are designed. Biosafety cabinets are primary means of containment developed for working safely with infectious microorganisms. So these are one part of biosafety program, biosafety cabinets. They require consistent use of good microbiological practices, primary containment equipment, and primary containment facility design. So to be more precise, BSCs are designed to provide personal, environmental, and product protection when appropriate practices and procedures are followed. So this is what I mean by BSC. And those are designed, or those are designed with the SOPs which are given by these two health organizations, CDC and National Institute of Health. Now, some brief historical perspective about the development or how this biosafety cabinets actually they are modified. First of all, when we were working with microorganisms, it started off with simple clean air cubicles. Clean filtered air was blown directly on the working surface inside the cubicle, like we all see now in IT companies and all. We see cubicles are given to each employee. So same way, it was a simple cubicle where fresh or clean air will be blown and there will be work performed on the working surface, and that's all. After that, concept of small workstation was came forward. So these are workstations which are bigger than cubicles and which are made up of wood and stainless steel. After that, ventilated cabinets were come, they came, and then now till nowadays, what we follow is HEPA filters. So high efficiency particulate air filters, they were introduced, and they still now they are getting modified, but it is standard. HEPA filters are used. Modifications are done with respect to designs, whether to use single or whether to use dual HEPA filters. We are going to see that classification. So first, let me tell you something about HEPA filter. HEPA filter is high efficiency particulate air filter. It removes the most penetrating particle size of zero point 0.3 micrometer. You can imagine how small it is, zero point 0.3 micrometer, with an efficiency of at least 99.97%. Typical HEPA filter. What is HEPA filter basically? It is a single sheet of borosilicate fibers and it is treated with weight strength water repelling binder. I'll show you this. I'll tell you first this points, then we will see the diagram. It will be more clear. Filter medium is splitted to increase the overall surface area. As we all know, basic principle of mathematics is if you want to increase the surface area, you can make folds into the structure. So filter medium is splitted in order to increase the surface area. And these splits are separated by corrugated aluminum tubes, folded aluminum tubes. So what is the purpose of this separation? Is to prevent the collapse, and it removes the particulate matter by three mechanisms: interception, impaction, and diffusion. Filtering efficiency depends on the fiber diameter, thickness, and phase velocity. You can see here, these are HEPA filters. So you can see here, this is the continuous sheet of fiber filter medium, and this you can see aluminum separators. As I told you, this sheet of filter medium is separated by pleats or uh, corrugating aluminum fibers or aluminum plates. So this makes the structure of HEPA filters. Now, what are importance of biosafety cabinets? Three important points are there: protection of personnel who is handling the infectious material, then protection of environment by preventing the release of microorganisms, and then protection of product as well, which are being handled. It's protection of personnel, environment, and product. So let us talk one by one. Uh, Biosafety Cabinet 1. It provides personal and environmental protection but no product protection. So in Cabinet 1, you are giving protection to personal and environment, but product protection is not guaranteed. Exhaust system is HEPA filter. What is the difference in all the cabinets that we are going to see? Class 1 PSC, unfiltered room air is drawn in through the work opening and across the work surface. And what is the minimum velocity of this airflow? It is 75 linear feet per minute. So always when you're talking about HEPA filter efficiency, you talk in terms of the what you can say uh, minimum velocity, and it is always in the unit of LFM, linear feet minutes. So minimum velocity of the airflow should be 75 linear feet minutes. What are the requirements? So what is the design? It is open fronted with the glass in the upper front. Integral tray is present to contain spills and splashes. Inward airflow is 0.7 to 1 meter per second. And protection factor has to be 1.5 to 10 is to 5. Protection factor meaning number of particles which, if liberated into the air of the cabinet, will not escape into the room. So that is how you are protecting environment, protecting personnel. So this is measured in terms of protection factor. And filtration from the exhaust air is through HEPA filter. This is the diagram of class 1 cabinet. If you can see, I've shown here one section view and one front view. So up at the top, you can see HEPA filter. Your room air is entering from here, right? So room air is entering from here, and this is contaminated air in the workspace. This contaminated air is going up and it is removed through HEPA filter into the environment. Wherever there is negative sign, it corresponds to negative pressure. Okay, so simple, right? Class 1 cabinet, room air is coming in, it is getting contaminated, and contaminated air is going out through HEPA filters. So when it is leaving the cabinet, it is filtered air, right? So this is the front view. Second, biosafety cabinet class 2. Uh, Purpose is the product protection. Laminar airflow, this is based on the laminar airflow principle which was put forth in 1960. There is, but here also still there is a risk of contaminant release into the lab and risk of product contamination. Now how to minimize this release is by means of two different designs. In class 2 also, we have two different designs, type A1 and type A2. So in type A1, you have internal fan, which draws room air at about 75 LFM velocity, and then supply air flows through the HEPA to the, which is a particulate free air to the work surface. What is the benefit? It reduces turbulence or disturbance and it reduces cross-contamination. So here, the downward moving air is getting split into two types, to the front green and to the rear green. 30% of the air goes to exhaust HEPA filter and 70% of the air recirculates through the HEPA filter back into the work zone. So understand, this is the basic difference. In biosafety level cabinet 2, always remember 30% goes to HEPA and 70% recirculates. Okay. Now, depending upon this ratio of recirculation and exhaust, there are different classifications. Okay, so this is 30-70. Now, when you talk about type A1, then there are two more points important. When you are working with class 2 cabinet, you are not supposed to work with the chemicals which are toxic. Okay, and exhaust air should be outside the building with the use of canopy hood and filter housing. They should not be ducted to the building exhaust system. Understanding? Now, this is how class 2A1 looks like. Your room air, see here, this is again a section view and front view. Your room air is coming inside, then potential contaminated air, and under positive pressure, it is going up and then it is released from HEPA filter. This is a section view, this is a front view. Are you getting? So here you have 30-70. 30% is released, 70% is recirculated. Class 2A2. Inflow air, here inflow air velocity was 75. Here it is increased to 100. All positive pressure contaminated plenums within the cabinet are surrounded by negative air pressure plenum. This is the basic difference. This is A2. Can you see here? There is a difference. There is no difference in the airflow pattern when you're talking about A1 and A2. But there is difference in the plenum. That means like in the A1, class 2 A1, always remember that the plenum which you are acting with is surrounded by the ambient air. Okay, and in class A2, class 2A2, the plenum which is under a positive pressure is surrounded by negative pressure. What will be the benefit of modification of this type of system? It would be, see here, what will happen because of this is in A1 and A2, you are changing the proportion or you are changing the circulation of air. So according to the ratio of the circulation and exhaust, you are dividing this into A1 and A2. When we are talking about A1, then we are talking about the circulation of air which is ambient and which is surrounded by the positive pressure. Here in A2, it is surrounded by negative pressure, because of which what will happen is there are risk of leakages which are completely eliminated when you are working with A2, because there is a presence of negative pressure. So negative pressure blower will take inside and avoid any leakage of the contaminants because they are under negative pressure. Then class 2 B1 and B2, when we are dealing with hazardous chemicals and carcinogens. Here, the ratio changes from 70 to 30. Okay. Class 2B1, you can see here, your room air is entering in. This is a dual HEPA filter. So, room air is entering in. It goes to HEPA filter again. And then it is this HEPA filtered air is coming up. And then it is going outside with the exhaust HEPA filter. Okay. So this is the section view and this is the front view of it. This is class 2 B1 and this is class 2 B2. In class 2 B2, what happens here, you can see room air is coming in, then it is completely going outside. Okay, so this portion is coming in, then from up room air is coming, from here also room air is coming, and both this what you can say flow of contaminated air is going up and it is getting exhausted through exhaust filter. So in B2 you have two types of, you can say, HEPA filters. One is supply and one is exhaust. Class 3. Class 3 is basically for highly infectious agents and hazardous operations. So there has to be care which is taken for it to be a gas type. There should not be leak greater than 1 into 10 raised to minus 7 cc per second. Cc means cubic centimeter per second with 1% test gas at 3 inches pressure water gauge. There, there is no opening view window. So for class 1 and class 2, you have opening air windows. Here you don't have opening windows. Passage of materials through a dunk tank and double door pass through box with autoclave. These are some conditions of class. Okay. Then, as I told you, it has supply and exhaust air. There is completely negative pressure cabinet. Okay, and there are long, heavy rubber gloves. Okay, heavy duty rubber gloves which are attached in a gas tight manner, so you don't have complete exposure to workstation. It is non-opening window, only there will be two openings to the biosafety cabinet and there will be rubber gloves attached. You have to enter your hand, work in the workstation. There is a negative pressure, so you are not allowed to enter any part of your body into the workstation. This is class 3. You can see here, these are the rubber gloves. So this is the front view. So this is one HEPA filter from where air is coming in and you are working in this working station and then it is going out. Okay. Yes. So now I believe that you are clear with class 1, class 2. Maybe briefly I can go through it once again if it is too much for you at a single dose. I'll just go through once again. So BSc 1, we have a simple exhaust system with HEPA filters. This is class 1. In class 2, we have A1, 30 and 70 is the ratio. So this is A1. This is the flow pattern. A2, positive planar is surrounded by negative. This is the most important feature. Or there is always a question asked, what is difference between A1 and A2? So main difference is that here positive pressure air is surrounded by ambient air and here it is surrounded by negative air pressure. So that makes or that avoids in class A2, it avoids the risk of leakage. Then B1 and B2, we have what is the difference between B1 and A is the ratio 70% and 30%, unlike 30 and 70. Okay, this is B1. This is the flow pattern. Then you have P2. Then P3 is completely different or at the topmost level of cabinet, which is very expensive to design. And here you are maintaining completely negative pressure with the rubber gloves which are provided in order to avoid any part getting exposed to the workstation. Fine. Okay, so this was about one part of biosafety, that is working with biosafety cabinets, which is very, very important. So now, as I mentioned in the earlier case, in the beginning itself, that coronavirus, Ebola virus comes in level 4. So which biosafety cabinet you should use? BSc 3, right? Now I am to talk about little bit about rules and some protocols which govern biosafety. So Cartagena protocol is very, very important, which decides the biosafety measures in biotechnology. It is the subsection of CBD. Okay, what is CBD? It is Convention on Biological Diversity. And I would give you one example for this. GMOs, that is genetically modified organisms, which and what is the biosafety measure for this? usually face challenges import and export. Okay, so imagine, or there is a situation where US produces this golden rice and India, we are importer of this rice. So when US wants to export this golden rice, then why it is important? Golden rice has high quality of starch. Okay, it is very, very good. Golden rice is a good modification of non. So golden rice is genetically modified. So now if US wants to export this GMO or golden rice to us, there are certain criteria or there are certain protocols which are to be followed, and that is known as Cartagena. So first US will apply and India will apply for import and export, and there is a what you can say office or authority which is biosafety clearance house. This biosafety clearance house will of both for both the countries. So whoever are the treaties which are whoever whichever countries are they follow CBD, they automatically they are like they become a part of they become a part of Cartagena protocol. Okay, so you have to follow Cartagena protocol. That means this BCH biosafety clearance house of they will read and analyze. They will do risk assessment study for about 270 days, and then it will be decided by BCH according to the Cartagena protocol whether to give license for import of this GMO to India and export of this GMO to US. So this is a normal procedure. Now for your better noting of and understanding of whatever we discussed, I put on this compilation of everything in one slide. You can see here biosafety level 1, 2, 3, and 4. Its description, no containment. So this is like a summary of whatever we have discussed in biosafety levels. No containment, BSL2 is containment, BSL3 is high containment, BSL4 is maximum containment, sample organisms being E. coli, BSL2 influenza and HIV, BSL3 tuberculosis, BSL4 Ebola virus, pathogen type. These in BSL1, they are the agents which present minimal potential hazard to personnel and the environment. They cause moderate into 3 exotic agents, agents that present potential threat, and BSL4 non-curable in most of the situations, very dangerous. Yeah, so I believe you all are clear with um, the things which we discuss about biosafety and bioethics. I hope it is like it was informative for you. Now, I'm going to go back to the bioethics which we discussed. You remember we discussed 4 principles. Based on that, yes, autonomy, beneficence, non-maleficence, and justice. Based on this, I'm going to discuss 2 case studies, okay, and 1 quiz. That first I'll tell one by one the case. Then let us make it interesting. You think over it, and I would like to get participation or views from uh, everybody. Everybody actually, but it's voluntarily. If you want to answer, you can answer. Yes. So first, I'll tell you what is the case all about. Carl is a 21-year builder. Okay. He's engaged to Julie, and she has recently discovered that she's expecting their first child. In 2001, Carl's maternal grandfather died from Huntington's disease, HD, which is a late onset degenerative disease of the nervous system. Now, you must have heard, I think, HD is inherited in dominant fashion. So, if you have HD, you have 50% chance of passing it on to your children. Now, Carl's mom has decided not to take the test to find out if she had got faulty copy from her father. But now that he's expecting to be a father himself, Carl is keen to find out if there is any risk that has passed on the condition. You understood the case? So, Carl and Julie, they are married, and Carl's maternal grandfather, he died of HD. Now, knowing the biology, 50% chances are there that it is passed on to the offspring. So Carl's mom, knowing this fact, she's not ready to get the test done. But now that Carl wants to become a father, he wants to know what is his genetic status. So whether so for that he wants the test to be done. Now there are some ethical issues at stake for Carl and Julie. What are the consequences of taking the test or deciding not? Now just think, if you were a Carl, what would you do? So see, if you analyze this scenario, Carl can know his status and prepare accordingly. But his mom has decided with the conscious decision that she does not want to know. So if Carl decides to take this genetic screening, there is a relevance of the decision or relevance of the fact that it can be a potential harm to her. So what are other consequences? Carl's relationship with Julie, Carl's relationship with baby, and implication for Carl's work. And some other issues like insurance implications, etc. Okay. So I would like to know, on the basis of four principles and your own conscience, what do you think Carl should do, and what will be the analysis of the situation and at the end? Okay. So anybody from your side? I would like team to unmute the person who wants to talk. I feel it should not go one way, and it's interesting always to know opinions of the experts and people who are in the same field. Even I would like to know your views, right? When I used to teach also, I used to take mute course and mock course of students and their opinions so that it becomes interesting, right? We get to know each other's views.
Thank you,
Anybody?
ma'am. Yes,
Yes,
ma'am.
Neeraj. Yeah, please go ahead.
Thank you, ma'am. Thank you. And thank you for allowing me to answer. As no one has taken this initiative, I would like to volunteer myself.
Yes.
Yes, I would like to go with the genetic screening.
Mm,
There are CVS or amniocentesis testing could be done to find out whether the fetus is affected with the 50% chance of getting the risk of HD gene transferred.
mm,
If yes, if say suppose worst case scenario, what would happen?
mm,
The gene has transferred, fine. Huntington
mm.
disease is not completely incurable. There are
Yes.
chances whereby we can maybe extend, or maybe we will get it late to late onset, and it is a late onset. It is not a early onset degenerative disease. So
Perfect.
so we can try to it's always better to be knowledgeable and prepared instead of taking like beating around the bush. If we are
Oh,
focused, we can very well take care of the baby. And
perfect.
other things like insurance and all, it doesn't matter. Life is
Yeah,
very
those are
important.
secondary. And what do you think? How should be the situation of his mom? Mom analyzed? Should
Uh,
she be given right under the any of the principle of bioethics? What should be that?
Sorry, ma'am, I couldn't catch the question properly. If you could please
How do you
little bit
justify? Yeah, how do you justify Carl's mom stand?
to not to go for the test.
Yes. Should it be allowed, or do you think health professionals should compel on it?
Not compelling, ma'am. We must make her aware that what are the worst case scenario and what are the best case scenarios. So I understand Carl's mom might be having some hesitation, maybe
Hmm?
due to lack of awareness. And even after awareness, if she is hesitating, then we must respect the Carl and Julie's decision because they are the parents of the
Right.
fetus.
Yeah.
So if the Carl and Julie they both give the consent unanimously, then we must go for the amniocentesis or CVS analysis, and we must find out the screening. In fact, I would like to go for some other diseases screening as well, which
Mhm.
might be running in the family. So a pedigree analysis must be done by genetic counseling, and then only we should proceed further with the proper counseling. It is quite possible to contain the situation very effectively.
Perfect. Fantastic, Neeraj. I'm
Thank
glad
you,
to
ma'am.
have participant like you.
Thank you, ma'am. Thank you.
May I know your profession? I think you're a doctor. Yeah?
No, ma'am. I am basically a medical geneticist.
Oh, fantastic. So you all conduct this kind of studies? Genetics.
Yes, madam. I actually, basically, I am a pharmacist. I did my diploma, bachelor, and master in pharmaceutical biotechnology from India, JSS. And later on, I went to University of Glasgow for my medical genetics studies. And afterwards, I am in different professions like research, academics, and all. Right now, I'm in Malaysia in AMS University.
Super. Fantastic to know about you.
Thank you, ma'am. Thank you.
Okay. Anybody else? I want some, if there are any student participants, I would like to know their view and their opinion. Mrinalini, do you want to contribute?
I also feel that knowledge is power. So, to that extent, I think if Carl carries out the test on the fetus using amniocentesis and checks whether the fetus has this faulty gene for Huntington's disease. I think the days ahead may be easier for them, but yes, one of the four principles which talks about beneficence, it talks about also non-maleficence, which includes the absence of coercion, and it also implies informed consent. So, like, no one can sort of force Carl in the
Super.
same
Yeah.
that his mother also cannot be forced to
Yes, yes, yes.
carry out the test. So, in that sense, I don't think that, these are things where, a third party really can sit in judgment.
Yeah.
Ultimately, it is left to the individuals themselves. So, informed consent and awareness is the need of the hour.
Yes. So basically, the bottom line is thank you, Mrinalini. Excellent. May I know what is your profession?
Yeah, I'm a doctorate in biotechnology from the University of Mysore, and I am an associate professor at the Sir M. Visvesvaraya Institute of Technology, Bangalore, an
Mhm.
engineering college, where among the subjects that I teach, among the courses that I have been teaching, bioethics and biosafety is one.
Okay, fantastic. It's such a great and wonderful experience to come up with the participants who have excellence in all these fields. So it's always a nice experience to interact. Thank you so much for your answer.
Thank you.
Yeah. So now the one more case study I'll just put in front of you. Wendy and Paul Carter. They have been married for 12 years. They would love to have children, but unfortunately, Wendy had breast cancer when she was 28. Although the chemotherapy has brought total remission from the disease, it also caused damage to her ovaries that has made her infertile. Paul and Wendy have been on the waiting list at their local IVF clinic for a number of months awaiting donated eggs to try and have a baby. At present, however, there are 2,200 potential mothers seeking each donated egg, and that the couple know that realistically they may never receive a donated egg via normal channels. Researchers at hospital attached to the IVF clinic have recently gained permission to carry out experimental procedures using eggs harvested from aborted fetuses. This technique is controversial, but for Paul and Wendy, it may represent that this is the only chance to receive a donated. So I think you have understood the situation. Wendy and Paul, they are married. They are desperate to have a children, and they got to know about the local IVF clinic where they are on the waiting list, and there are many other mothers also seeking for the donated eggs. But the what you can say uh, technique is controversial. Technique of such kind of conception is controversial. So whether this Wendy and Paul should go for this controversial technique and try a luck, or even if it is not giving any guarantee, so should they try a luck, or they should not undergo such technical controversial treatment being unethical? What do you think they should do?
If I may just
Yeah,
ask
sure.
a
Yeah.
question. The issue that is in focus here is the question of abortion. I mean, it's using aborted
Yes, 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 yes.
fetuses, that is, eggs harvested from aborted fetuses,
Correct, correct, correct.
for which we need more number of aborted fetuses in order so that the 200 potential
Yes, yes,
parents are enabled. That is the problem that is here.
yes.
Of course, I mean, so ultimately, I think this case study boils down to the fact that do you support abortion or otherwise? That is one of the questions that I see through this particular case study.
Hmm.
According to me, I mean, I think it may be necessary. I would say, yes,
They
aborted
should go ahead.
might increase in case
Hmm.
uh, abortion definitely would be needed if there is a threat to the life of the mother, or if the lady is a victim, or various other factors that necessitate what is termed as a medical termination of pregnancy.
Yes,
In such
correct.
cases, definitely. Of course, we do not know whether the numbers would, enable all 200 the potential mothers, including this couple in question. So the thing is, I think they can try, but uh, again, if we were to advise them, if they were to ask any third party's opinion, I would say that, well, they cannot, the chances they have to consider, what is the percentage chance of success? They should be informed about it, and ultimately the decision is theirs. That's how I would put it. depending upon the chance. The technique is controversial, but it may represent their only chance to receive a donated egg. What is the percentage success? The pros and cons. They have to be informed about it, based on which the decision is theirs, whether to go.
Perfect.
Also, one more thing is there, since Wendy has already had a serious illness, and there is a, problem that she's recovering from. It also brings in that angle. I mean, it was only with the doctors, I mean, her doctors who treated her for her malignancy, they would also come into the picture here.
Yes. So here multiple, all those four principles have to be thought at a time. And then the wise decision has to be made on the basis of four principles of bioethics. You're
Autonomy
right.
predominates, and by way of informed consent. Also, as far as the healthcare specialists are concerned, the question of beneficence and maleficence, etc., those also come in. Yeah.
Yes. Fantastic, Mrinalini. Thank you so much for your answer and participation. Excellent. Neeraj, you want to give some input?
Sure, ma'am, but I need a little clarity on something.
Yeah.
Uh, the experimental procedure using eggs harvested from aborted
Mhm.
fetuses,
Mhm.
is it
Yes.
a reversal of the process, or are we talking at the blastula or gastrula state of the unutilized zygotes?
Unutilized cycles.
Okay. Whatever Mrinalini ma'am has mentioned, I do agree
Mhm.
with that. However, there
Yes.
are two, 3 points which I think should also be considered.
Mhm.
If the IVF clinic is going to, in collaboration with the hospital researchers, definitely are going to provide these eggs of unutilized fetuses, that means these are not of Wendy and Paul for sure. One thing. So we must have a consent from those
Yes.
parents who
Mhm.
had been the actual parents for these unutilized eggs. So in that case, do we have the consent from them?
Yeah, so that, but then Neeraj, it comes as a part of SOP which is followed by the hospital. So they, first, it is a priority thing. So they must have, or we have to assume that they have got a consent because since it would be in the clinic available. So the parents, they must
Yes,
have by their own willingness, they must have donated
yes,
it,
ma'am. But
right?
my personal experience
Hmm? I can't hear
indicated
you.
something
Haha, yes,
else.
yes, yeah.
Like
Hmm?
my personal experience related to this little bit of work which I did. So,
Yeah.
according to that, those who got help from IVF clinics,
Mm-hmm.
the closest, definitely the country to country the situation will change. But
Yeah.
those who are seeking this kind of help, they would make sure that the unutilized eggs will not be utilized by anyone else, or will be kept in cryo for a longer time duration in case if they need it in future. So the
Mm-hmm.
clinic cannot dispense these eggs to any another seeker or any another person in the waiting list without their consent. So
Mm-hmm.
that's one point which, as per me, must be taken consent from them. One thing. Secondly,
Uh-huh.
ma'am, uh, they are married for 12 years and there are
Yes.
200 potential mothers. If not out of those 200, I believe
Mm-hmm.
that they are also from those 200 number of waiting list members. So if 199 people are not going for this particular controversial experimental procedure, why
Yes.
are they choosing? What are the risks associated with these for
Mm-hmm.
Wendy, especially? As Manali madam has mentioned, she had been
Mm-hmm.
treated for breast cancer very early. Plus, she got her ovaries affected with that. Is her body going to support the particular process? Definitely, they must be going through with these assisted reproduction support members and all. but that aspect is also required. So I do agree with Mrinali ma'am's point here, ma'am.
Yes, yes,
Thank you,
yes.
ma'am.
Fantastic, Neeraj. Super. Very well analyzed by Neeraj and Runalini. Excellent. That gives me all the satisfaction and pleasure of designing this module. Last, very simple quiz for all. Uh, After a motor vehicle accident, a patient was rushed to emergency room where splenectomy was performed. Splenectomy means spleen is removed, and this is because of damage for the accidents without the patient's consent. How do you justify this? Autonomy, beneficence, non-maleficence, or justice? You get the question?
Yes, ma'am. Let others try the answer first.
Okay. Pooja, can I have your answer? Sakshi?
Yes, ma'am.
Yes, Sakshi, please go ahead.
Uh, I think it can come under beneficence because as he needs to be saved, so I think the doctor must do everything which from whatever possible way that they can save him.
Perfect. Your answer is right. So I believe everybody thinks on the same line. Neeraj, you had same answer, right?
Yes, ma'am. Correct. Absolutely correct.
Perfect. Superb. So I would like to end this talk with the vote of thanks. You all have been very patient and excellent participants. Neeraj, Pranalini, you had a nice interaction with me. This is
Yes,
the
ma'am. Thank you
first
so
time
much, ma'am.
yeah, most welcome. This is the first time I did this experiment because whenever I conduct the seminars and workshops, it is always a one-way talk. So, but today I thought of interacting with participants. Usually it's on my other expertise like bioinformatics. So everything was one way. But today I enjoyed it a lot because you were responding so well. So thanks a lot. And please visit our website. And I think team must have provided in the chat box all the links where you can uh, visit our workshops. Any help, anything you need from this field, we are more than glad to help you. Okay. Thanks a lot. And I'll end the meeting here.
Thank you so much. It's so pleasant to hear a workshop on a relatively less explored area like bioethics and biosafety. It must
Yeah,
be
yeah,
very
Correct. Correct.
interesting to your
Correct.
organization.
yeah. I
Shar
think, yes.
Sharwari ma'am,
Yes, Neeraj.
sorry to interrupt in between. I
No.
attended your bioinformatics workshop the other day, and
Okay.
I would like to admit, just due to that one, I'm sticking with BDG Life Science, and I'm trying
Thanks
to
a
attend all.
Such a big compliment for me, Neeraj. Thanks
Yes,
a
ma'am.
lot. So always be in touch with me
Sure, ma'am.
and
Sure.
with BDG team. We would like to give you projects also. You can go through website and
Yes, I know. I know.
there I have my mail address also, so you can be in contact. In fact, we can be in contact. It's a vice versa procedure
I
always in
understand.
the
Yes, ma'am.
education field. You will also miss. I also get to know something from your side.